Welcome to the opening lecture of the Master on Integrative Synthetic Biology, an initiative of the National Research Council, CSIC, and the International University Menendez Pelayo. We officially start the first edition of this master with the inaugural lesson by Jack Szostak, professor at Harvard University and researcher of the Howard Hughes Institute, now moving to a new scientific adventure at the University of Chicago. Good morning, Professor Szostak, and good afternoon to all of you attending the lecture from Europe. My name is Juli Peretó. I am professor of biochemistry at the University of Valencia and member of the Institute for Integrative Systems Biology, a joint center of CSIC and the University of Valencia that together with the National Center for Biotechnology, CNB, and the Center for Biological Research, CIB, are the organizer of this master. This event is also part of the activities of the CIC Conexión Vida network recently created on the basis of the CIC Challenge Origins, Coevolution, Diversity, and the Synthesis of Life. I will chair this session together with Kepa Ruiz Mirazo from the University of Basque Country on behalf of the director of the master, Professor Germán Rivas. We all, teachers and students, wish to express our gratitude to Professor Szostak for agreeing to deliver this inaugural lecture. I will now give the floor to Carlos Briones, researcher at the Astrobiology Center in Madrid, who will offer a biographical introduction of our invited speaker. Carlos.
Thank you very much, Julie. Good afternoon, everyone here in Spain. Good morning, Jack, in the States. Well, it's my pleasure to join you today and to introduce Professor Jack Szostak, who is Nobel Prize in Physiology and Medicine in 2009. As many of you know, Jack Szostak is Professor of Chemistry, Chemical Biology, and Genetics at Harvard University. He's a senior investigator of the Howard Hughes Medical Institution, and he's also the Alex Rich Distinguished Investigator in the Department of Molecular Biology at the Center for Computational and Integrative Biology at Massachusetts General Hospital. And now he's moving to Chicago, as Julie announced it. Jack's early research on telomere structure and telomerase function and the role of telomere maintenance in preventing chromosome degradation and cellular senescence has been recognized with numerous awards, including, of course, the 2009 Nobel Prize in Physiology or Medicine, shared with doctors Elizabeth Blackburn and Carol Greider. In the decade of 1990s, Professor Szostak and his colleagues developed the first experimental protocols for in vitro selection of information-bearing molecules in the test tube, thus allowing the isolation of functional RNA, DNA, and also protein molecules from a large pool of random sequences. Indeed, in a seminal paper published in 1990, together with Andy Ellington, they coined the term aptamers, one of my favorite ones, that we use in this field to refer to in vitro evolved target binding nucleic acids. Current research in Jack Szostak's lab include the experimental system of self-replicating protocellular systems as a bottom-up approach to the origin of life with groundbreaking contributions to this field. And he is particularly interested in using systems chemistry-based approaches involving complex media with increased molecular heterogeneity. Well, I met Jack Szostak more than 20 years ago in the framework of one of those ESOL meetings about the origin and evolution of life. And I can tell you, students, that apart from being one of the most relevant researchers in these fields I have referred to, he's a brilliant speaker, and he's also a very warm and friendly person. Indeed, I will never forget, Jack, your enlightening talk on the origins of life on Earth at the Ramon Areces Foundation and Real Academy of Sciences Joint Symposium 5 years ago here in Madrid. So it's a pleasure to have you at that symposium, and it's a real pleasure for all of us to have you here. So thank you again, Jack, for joining us, and the floor is yours.
Great. Thank you very much, Carlos, for your very kind introduction. Thank you also, Julie and Kepa and Germán, for inviting me to speak here. And thanks for all the students for coming to listen. And I'm also looking forward to questions later. So let's see, let me share my screen and we can start here. Okay, I hope everyone can see this. Okay.
It's perfect.
Good. So, so what I wanted to talk about today was our efforts to understand how the chemistry on the early Earth gave rise to the beginnings of life, and in particular, how the first very primitive cells or protocells emerged from that chemistry. So our general approach is synthetic, in the sense that we're trying to see how we can build structures like this in the laboratory, and in trying to do that, we get clues about what kinds of pathways are possible and what are not, and thereby we gain some understanding of what the possible pathways to life might have been on the early Earth some 4 billion years ago. So the image that you see in the background of this slide is a schematic version of what we think a very simple protocell would have looked like. And it's really a simplified version, but the two components of such a cell that I'm going to talk about mostly are inside. You can see small fragments of some genetic material, which we think is probably RNA or something like RNA. So I'm going to talk a lot about that. And these fragments are encapsulated inside a membrane bilayer made out of very simple molecules. But you can see that the overall structure is similar to what we have in biology today, cells with a membrane boundary, genetic materials on the inside. Of course, modern cells, as I'll talk about a little bit, are much more complicated, and we have a lot of machinery and a lot of proteins. So we didn't have enzymes or coded protein synthesis at the beginning of life. So the real puzzle is to understand how very simple structures like this could self-assemble and also could grow and divide and replicate, and most importantly, begin to evolve in a Darwinian sense to adapt to their environment and in so doing become gradually more complex until eventually we ended up with modern life. Okay, so I'm going to start off by talking about these genetic materials, which, as I said, we think are RNA. And one of the questions is, why do we think that life started with RNA and not something else? But to begin talking about RNA, I think these days it's almost impossible not to mention the little piece of RNA that has affected us all so much in the last 2 years, and that is the RNA genome of the SARS-CoV-2 virus, the cause of the COVID-19 pandemic. And I think this is just a really amazing example of the power of this simple genetic material. So the genome of this virus is a roughly 30 kilobase stretch of RNA, and it encodes this fairly large number of proteins that do all of the viral functions and enable it to take over cells that it infects, and has the power to spread across the world and basically almost bring the whole world to a standstill. And that's because of this little 30 kilobase piece of RNA. So if you think about the amount of information encoded in that, it's less than a 10 kilobyte text file. I mean, there probably are no Word files on your computers that are this small. So it's just an example of how amazing RNA is. So this is the RNA that has caused so many problems. But then what's also happened recently, and it's been incredibly dramatic, is RNA has come to the rescue through these new mRNA vaccines. And you probably know that basically what's going on here is that messenger RNAs are generated in the lab on a very large scale these days, encapsulated within a simple lipid nanoparticle. And these particles that make up the vaccine can then enter cells in a way similarly to how viruses enter cells. Then the RNA is released, translated, makes peptides and proteins, which stimulate the generation of antibodies as well as B and T cell responses, and give us immunity to the virus. So again, a testament to the power and the importance of RNA in biology. Now, of course, if we look at modern cells, we see RNAs everywhere. RNAs have so many roles in modern cells. And this is one of the reasons that we think RNA was probably important going all the way back to the origins of life. So we see RNA molecules playing roles in the generation of peptides and proteins in multiple ways. So there are the large RNAs that are the main components of ribosomes. There are smaller transfer RNAs that bring amino acids to the ribosome. There are microRNAs that regulate RNA translation. There are riboswitches that again play regulatory roles. And even if you look at many of the coenzymes that are important for metabolic enzymes, a lot of them have little pieces of RNA, basically adenosine as part of these cofactors. And so many roles for RNA in modern cells really makes us think that RNA had a very ancient origin. And perhaps the best and most convincing example of that comes from the structure of the ribosome itself, the macromolecular machine that's found in all cells on Earth that generates proteins. And so this is an image from a crystal structure of the large subunit of the ribosome from the Stites lab many years ago now. And these gray squiggles are RNA. These gold areas are ribosomal proteins. And the green segments in the very center are the active site of the ribosome. This is where peptide bonds are made. And what you can see is that that active site is composed entirely of RNA. So the conclusion from this structure is that basically in all of life, everywhere on this planet, RNA molecules are what make proteins and peptides. And so logically, the conclusion from that is that RNA probably came first, and RNA somehow evolved the ability to make proteins. Proteins, of course, are almost highly dominant in biology today in terms of structure and function. But it appears from the multiple roles of RNA and from structural evidence like this, that the ancestral state, the early state, must have been a kind of biology dominated by the presence of RNA molecules. Okay, so if we look at the organization of modern cells, and here I'm talking about everything from bacteria and archaea to eukaryotic cells, all the way up to human cells, we have this common organization at the informational level. So in cells, we have information is archived in a DNA genome, which is transcribed to make RNAs, both messenger RNAs and all of the various regulatory and structural and functional RNAs I just mentioned, which are then translated to make proteins, which do most of the jobs in the cell of structure and function. So if you think about this kind of organization, you can see why it made thinking about the origin of life so difficult for so many years. And that is that every part of this depends on every other part. So you can't replicate DNA without enzymes, which these days are proteins, without RNAs, for example, for priming, and of course, without all of the metabolism that generates the building blocks of DNA. And the same argument applies to RNA and to proteins and the metabolites. Everything depends on everything else. And so how could such a self-referential system get started? It was really a major stumbling block to thinking in a logical way about the origin of life for many years. Now, it happens that in the late 1960s, there were a few super smart people like Crick and Orgel and Woese, who realized that there was a solution to that, and that lies with this molecule in the center here, RNA. And the reason that these people thought that maybe RNA was the key to the origin of life was that was the time at which the first structures of transfer RNAs were solved, three-dimensional structures. And people realized for the first time that RNA could fold up into complicated three-dimensional shapes. And so that led to the hypothesis that maybe RNA could also act as an enzyme. It was known even back then that RNA could act as a genetic material, both as a messenger RNA and as the genome of certain viruses. But if RNA could also act as an enzyme, then you could imagine much simpler cellular organization in which RNA played both of these roles. And you could then imagine DNA and proteins coming along as a later stage of evolution. Now, nobody really took that seriously for about 15 years or so until Tom Cech and Sid Altman discovered from laboratory studies that yes, in fact, RNA molecules can act as enzymes. They can have folded structures, they can have active sites, and they can catalyze chemical reactions. So that led to a much simpler conception of the origin of life. And so I've kind of drawn that out schematically here in essentially another version of what you saw in my opening slide. So again, we're thinking of very, very simple early cells with probably small RNA genomes encapsulated within some kind of simple membrane envelope. And so now we have a much simpler problem, but it's still, it's complex, and it's been keeping a lot of us busy for many, many years now. And that is thinking about how could structures like this spontaneously assemble. That turns out to be not so hard, but the really interesting and tricky problem is to think about how these kinds of structures could replicate. So for the cell membrane, that means how can these membranes actually grow and how can they divide, all without any pre-evolved machinery, right? There are no enzymes, no proteins, no cytoskeleton. Everything at the beginning had to be driven by simple environmental forces, the physics and the chemistry of the environment. And perhaps the most tricky problem of all is trying to understand how a genetic material like RNA could actually get copied and replicated so that it could be propagated through a cycle of growth and division. And most importantly, begin to accumulate information about the environment. In other words, the information that is accumulated as a result of Darwinian evolution is coded in these genetic molecules and expressed in the form of structural or catalytic activities that allow the cell to survive better, to propagate better, and so on. And so that information you can understand as information about the environment and about how to deal with the environment. Now, because this cellular organization is so simple compared to modern cells, I think it's very important to realize that these cells are highly dependent on their environment. The environment has to supply all of the building blocks, the nucleotides, the lipids, the sources of energy. And there are many sources of energy, right? We shouldn't limit ourselves to thinking of any one, because we have energy from the sun, we have ultraviolet energy, we have mechanical energy, we have various forms of chemical energy, and so on. And so the trick, the goal of all of our studies is to understand how structures like this could emerge spontaneously and start to evolve. Okay, so let me begin by talking in a little bit more detail about RNA. And so here's the chemical structure of RNA drawn out as a single strand and illustrating the 4 bases, the A, C, U, and G, that are used to encode information in sequence. And the structure of RNA includes the nucleobases that provide this informational identity and a backbone that's comprised of a sugar linked by a phosphate to the next sugar, and in a very homogeneous manner and it can be, as you've seen, in RNAs that are very long. Although at the beginning, we imagine these RNAs to be relatively short. So this is a pretty complicated molecular structure. And it's actually taken decades for people to understand how the chemistry of the early Earth could possibly give rise to molecules like this. And so I don't have time to go into a lot of the details of that chemistry, but many people have contributed, and I think I would highlight especially the work of John Sutherland in the UK in understanding how the basic nucleotide building blocks of RNA arose from very plausible chemistry, beginning with simple building blocks like cyanide and going through very elegant and high-yielding pathways to generate the nucleotides that make RNA. Now, thinking about the building blocks of RNA, there's an important aspect that was actually understood a long time ago, almost 40 years ago now, by the late Leslie Orgel. And that is, again, a contrast between modern cells and primitive cells. So in modern cells, where we have very fancy, very effective enzymes, we can use molecules like GTP, ATP, as shown here, that are, in a sense, activated nucleotides because of this triphosphate. So it carries a lot of energy, but it's kinetically stable. And you need a very good enzyme to make use of molecules like this. So if we're thinking about the origin of life, where you don't have enzymes, we need molecules more like what you see down here on the bottom. So these are activated in a different way with imidazole groups, shown here, methylimidazole. We use a slightly different version now. So these, again, are high energy compounds, but they're much more reactive. And so they can actually start to react and copy template sequences without enzymes. But learning how that process happens and learning how to make it more efficient has actually been quite an adventure, especially for us over the last 5 or 10 years. But so I'll talk a little bit more about that in a minute. But one of the puzzles I wanted to address in the talk today is, why did biology end up using RNA? And if it started with RNA, why did it start with RNA and not with any of actually quite a large number of possible alternative nucleic acids? And what I've drawn here are 2 alternative possibilities that are structurally and synthetically very close to RNA. So over here, we have arabidonucleic acid. And you can see from this diagram, the only difference is that this hydroxyl is pointing down in RNA and pointing up in arabino Na. Over here, we have 3O-nucleic acid, where the sugar it's similar, but it's 4 carbons. And in a sense, this looks easier to make than the 5-carbon sugar in RNA or ANA. So what are the factors that led biology to start, or at least to end up, with RNA and not with ANA and TNA? Okay, so again, just to look at this from the point of view of the building blocks, the nucleotides, here is an activated ribonucleotide that will polymerize to form RNA. And here are the building blocks of ANA and TNA. So they're very similar, except for these very small differences. And this is a puzzle that was actually recognized, again, decades ago, that in prebiotic chemistry, where the reactions are not guided and channeled by enzymes, it's just not realistic to think of making 100% pure ribonucleotides. There are going to be side products that are closely related from the chemical pathways that we're really beginning to understand. And so 2 of those side products are this ANA building block and this TNA building block. And so if you had a big mixture of all of these things, that just seems like it should be a problem. And so it leads to this question about, why did we end up with RNA? So you can think of the problem like this. If the prebiotic chemistry is not perfect and actually generates a lot of different products, so maybe mostly ribonucleotides, but certainly there will be some of these variant nucleotides, so arabino, 3O, even deoxynucleotides, the building blocks of DNA. So if they're all mixed up together in that nice warm prebiotic pool that maybe gave rise to early life, and these start to join up with each other to make short chains, they'll be very heterogeneous, extremely messy. And it's hard to imagine how this kind of messy mixture could actually give rise to something more homogeneous that would be a better starting point for evolution and for the generation of functional folded molecules, which is really what we need to allow cells to evolve to become better adapted to their environment. So this is a puzzle that's been recognized for decades. And so for the next few minutes, I want to talk about a possible solution to how, even though you start with a kind of a mess, there's a simple way in which you can end up with more or less homogeneous RNA, something that looks much more like modern RNA. Okay, so to understand that, we have to think a little bit about the chemistry of copying. And so what we've been doing in my lab for the last 5 years or so is trying to understand if there are differences in the copying chemistry that might explain why RNA won out in the end. So the way that we think about RNA copying is shown here. So our kind of more classical model is based on what we see in biology, where you have a template strand, you want to copy the information so that you can get replication started. And that works by having a primer sequence that is extended 1 unit at a time by reaction with activated monomers, such as I showed you. Now, in terms of origin of life, it's quite likely, I think, and this is just a hypothesis though, that the beginnings might have been a little bit more heterogeneous. So maybe copying actually worked by some combination of having primer extension, building up short oligonucleotides, which could then join together with each other by ligation, and thereby build up a copy of a template sequence. So either way, we have to understand the chemistry and figure out how these reactions actually work. And So I'm going to walk you through that process. Don't worry too much about these chemical structures on the right, if that's because there's a simplified schematic diagram on the left, which is probably more important to pay attention to. So this has actually been one of the most surprising discoveries that I've had in my career in science. Because for many decades, we, and I think everybody in the field, assumed that template copying of RNA in this kind of prebiotic model worked more or less like copying works in modern biology. So we have a primer bound to a template, monomer comes in, reacts with the primer, and grows. What we found, or I should say, Travis Walton, when he was a graduate student in the lab, found is that it actually works in a surprising and different way, which is that two different activated nucleotides first react with each other to make this bridged intermediate. Chemical structure is shown over here. It's a surprising and unusual structure. But this bridged dinucleotide binds to the template. And the beautiful thing is that this bridge structure pre-organizes the three-dimensional shape so as to favor the chemical reaction with the end of the primer that results in this nucleotide shown in blue here, adding to the end of the primer. So we have primer growth by one nucleotide. And then the downstream nucleotide is released and can dissociate, and then you can repeat the whole cycle. So a very surprising chemical mechanism for template copying. The important thing in terms of understanding, why RNA and why not something else, is that now that we understand this process, we can ask, well, how would it work if we replace these incoming monomers with, for example, arabinonucleotides or threonucleotides or deoxynucleotides? Or we can ask, well, what if we put one of those variant nucleotides at the end of the primer or in the template? We can experimentally look at all these different situations. And so we can also look at this in really atomic detail by solving crystal structures of the various stages of this chemical process. And so what I'm showing here is a series of crystal structures that were determined by Wen Zhang when he was a postdoc in the lab. And we're just seeing part of what's an overall RNA duplex with a little template overhang, which is off the top of the screen up here. And what's been done is to soak in these activated molecules into the crystal. And at a short time, we see the activated monomers bound to the template strand. The activating imidazole part is disordered, so we don't see electron density. But if we wait, we see the formation of this bridge joining these two nucleotides. And at that stage, the 3' hydroxyl at the end of the primer is just poised to attack this phosphate next door to it and to catalyze formation of a new phosphodiester bond, which you see in the structure at the bottom. So by waiting for a few hours, we can actually see in this series of crystal structures the growth of the primer. So because of a lot of kinetic and structural studies like this, we now feel that we have a pretty good understanding of the basic copying chemistry. And so now we can say, well, what happens if we try to do the same thing with some of these other relatives of ribonucleotides and RNA that we expect to have been around due to the imprecise nature of prebiotic chemistry? And so a lot of this work has been done in my lab by many people, but a lot has been done by Chris Kim, a recent graduate student in the lab. And so in this particular experiment, what you can see is that we're putting in arabinonucleotides, and we can measure how quickly do they form this bridged intermediate. If we put one at the end of the primer, what happens? If we put one in the template, what happens? And basically what we see from actually measuring all these steps is that this bridged dimer forms perfectly normally, but it reacts 10 to a 60-fold more slowly than if this was the RNA version. Now, it's a slow reaction, but if it does react, you end up with A and A at the end of the primer. So we can make those synthetically. And that is really bad. It's 100 times slower than if this is an all RNA primer. The only thing that works decently, actually, is putting one of these variant nucleotides in the template. And that slows things down a little bit, but not too much. And again, we can use X-ray crystallography to try to understand why are we seeing these differences. And so here, this is a crystal structure that was obtained where we have this variant nucleotide, the arabinonucleotide, at the end of what would be the primer. And you can see that this 3' hydroxyl that has to react with the adjacent phosphate is much further away than when everything is made of RNA. So we have a structural explanation for why RNA works better in this case. And we can do similar experiments with TNA, starting with 3' nucleotides that we make, or rather our collaborator, John Chaput, makes. It's a little bit tedious to prepare in the lab, but it can be done. And this time we see that actually making this bridged intermediate is in fact a little bit slower. If you look at the rate of primer extension, that's also slower. If you put one of these nucleotides at the end of the primer, that's also slower. So every step along the way is slower with TNA than it is with RNA. Again, only putting it in the template has a relatively minor influence. Once again, of course, we want to not just see these kinetic measurements, but we want to understand what's going on. And this is fairly easy to understand because the difference between TNA and RNA lies in the sugar. And essentially, the phosphate is held over the sugar in TNA, whereas held out away from the sugar in RNA. And again, the result in the context of a duplex structure is that the end of the primer is much further away from the phosphate that it has to react with than in the case of RNA. So all of these kinds of experiments together show that essentially in a competition, RNA always wins. In fact, everything that we've looked at so far, and granted there are many more things that we have to explore, but so far, everything that we've looked at, RNA is better at copying chemistry. So if you put that all together into a model, this is the way we're thinking about it now. So again, we start with this somewhat messy mixture, which is due to the lack of precision and control in the prebiotic chemistry. This results in the assembly of short chains, short oligonucleotides that are very messy mixtures. They contain all of these variations that I've listed here and probably many, many more. But their sequences of nucleotides are also random. And this allows pairs of these chains to sometimes pair up by Watson-Crick base pairing. And this is the kind of geometry that you need for primer extension. So in the presence of activated nucleotides, this chain can grow in this direction, the chain on the bottom can grow towards the left. And from what I just showed you, we know that when these chains grow, it's going to be mostly ribonucleotides that get added and RNA that's going to be synthesized. And if you imagine this happening over and over again in many cycles, the chain's always growing a little bit, then coming apart, sometimes fragmenting, and continuing to grow by this kind of mechanism, after a while, what you'll have, we think, is mostly RNA, something that is maybe not perfectly homogeneous like RNA made by modern enzymes, but pretty good, much more homogeneous RNA strands that we think would be a much better starting point for the evolution of functional RNAs. So one of the things we're working on a lot these days is understanding not just copying chemistry, but the whole cycle of replication. And also how can that replication process happen inside these membrane compartments. Okay, so that work is ongoing. I'm going to sort of skip over that, but I'm happy to answer questions about it later and talk now again about the membrane compartment, because that's the other important half conceptually of our idea of a simple protocell. So what are these simple membranes and how do they form and how do they grow and how do they divide? So what we think is that primitive membranes were composed of much simpler molecules than what we have in biology today. And so we like to play around in the lab with simple fatty acids, such as shown here, oleic acid, or these shorter chain fatty acids. And the work on forming vesicles out of these simple molecules actually goes back way before I got involved in the field to early studies by Dave Deamer, Pierluigi Luisi, and others. But these molecules, under the right conditions, will spontaneously self-assemble to make membranes, bilayer membranes, which close up and make these incredibly beautiful vesicles, such as you can see in the microscope image on the left. Now let's look in a little more detail at the properties of these vesicles and why do we actually think that, that these would be better for the origin of life than more complicated membranes. And the answer has to do, I think, with some of the phase behavior of these molecules. In other words, what kinds of aggregates do they form spontaneously? So one of the really beautiful things about these simple fatty acids is that at high pH in alkaline environments, they make really tiny aggregates called micelles. And that's because the carboxylates are ionized and you keep the negative charges as far apart as possible while allowing the hydrophobic chains to self-associate. So that's the low energy form in an alkaline environment. But then if the pH starts to drop and get closer to neutral, then when you have roughly half of these carboxylates ionized and half protonated, they can hydrogen bond to each other and make this beautiful bilayer membrane structure that forms sheets that close up into spherical vesicles that you can see in the background micrograph. And it turns out that this allows us to feed preformed vesicles with new material. So we can make vesicles at lower pH and feed them with micelles at high pH. And we can actually watch these vesicles grow. And so the first time we were able to see this was from a series of experiments that were done by Ting Zhu when he was a graduate student in the lab many years ago now. We knew that growth worked in this kind of feeding paradigm, but we'd never seen it in the microscope until Ting joined the lab. And so I just want to show you a few images because this was another incredibly surprising result. And I think it epitomizes the fact that working in this field of origins of life, it's really a goldmine for discovery of surprising physical phenomena. You really just could never imagine some of the surprising things that can be uncovered by very simple experiments in this field. So what you're seeing in this image is a whole bunch of fatty acid vesicles, all about 4 microns in diameter and filled or loaded with a fluorescent dye, which is what you're actually seeing. So what Ting did was to make vesicles like this and then feed them with more fatty acids in the form of micelles, which then, as they go into this lower pH environment, they become unstable and those molecules enter into and allow the fatty acid membrane in the vesicles to start growing. So we thought they would just get a little bit bigger. And we did not expect what actually happened, which you can see here. and which is totally bizarre and still, after many years, a surprise to me. We can see these little thin membrane tails growing out of each vesicle. This is after about 5 minutes. And then if you wait for about half an hour, every spherical vesicle that was there at the beginning is now grown into a long, thin filamentous vesicle. And I should emphasize that the contents are still inside. That's why we're seeing it through the fluorescent dye. So nothing has leaked out, but the shape of the vesicles has totally changed. And the beautiful thing about that is that if you shake these up, these filaments are very fragile, and they divide into daughter vesicles. And that allows us to experimentally implement a cycle of growth and division that you can see here. So we start off with spherical vesicles. We add food in the sense of more fatty acids. They get incorporated, so the vesicle grows into a filament. Then we agitate, divide into new spherical daughter vesicles, which can grow again and divide and grow and divide. And you can keep doing that as long as you like. So we have a cycle of growth and division. Now, that's very nice, but that cycle does require the periodic addition of new food molecules, right? New fatty acids. And so we were thinking, wouldn't it be nice if you could also have growth in a more steady state environment? This would be maybe a somewhat simpler way to imagine it happening if you didn't need to have periodic introduction of new material. And so our first insight into how that might be possible came from work that Irene Chen did when she was a graduate student in the lab. And what Irene did was to prepare fatty acid vesicles that were loaded up with a lot of stuff. And that could be anything from sugars to nucleotides to RNA molecules. And the result is these vesicles are, because of the osmotic pressure, they're swollen. And as a result of being swollen, the membrane is under tension. So it's a kind of high energy state, but there's no relaxation pathway. So nothing particular can happen unless you introduce into the population relaxed vesicles that are not swollen, because they are isoosmotic. They have less material on the inside. When you do that, because these fatty acids are so dynamic, they can leave this membrane and enter the membrane of the swollen vesicle, which can then relax by growing. So the swollen vesicles grow, the relaxed vesicles shrink, and this is a pathway by which growth could occur in a competitive manner. So we were very excited about this, because if we thought that the osmotic swelling was due to faster replication of RNA molecules in certain vesicles, then better RNA replication would immediately be coupled with growth of the membrane compartment. Okay, so you don't need any fancy biochemical pathways. There would be a simple physical phenomenon that couples RNA replication with membrane growth. And I still think that's a very beautiful aspect of this. The problem was we weren't really able to think of good ways to divide osmotically swollen vesicles. They're quite stable. It takes a lot of energy to change their shape and make them divide. So our next insight into how that might happen came from a totally different direction. And this was work that was done by Itai Büden when he was a graduate student in the lab. And Itai was actually thinking about a different problem. We were imagining primitive membranes as made from these very simple building blocks, fatty acids, which have all the right kinds of dynamic properties, the permeability to building blocks like nucleotides that you need in a primitive cell. But modern cells, shown at the bottom right, have membranes made from more complicated molecules like phospholipids, And these are fantastic barriers. They're very impermeable. They're very stable. These molecules are locked into the membrane. And all of the dynamic behavior in modern cells is catalyzed by proteins. So Itai was thinking about, well, how could you have an evolutionary transition going from these very simple dynamic membranes through some intermediate state to the more stable modern membranes? So the logical intermediate would be a membrane composed mostly of fatty acids, but maybe with a little bit of these more complicated molecules like phospholipids. So the problem is, why would this evolutionary pathway actually happen? I mean, it wouldn't unless there was a selective advantage to these intermediate states. And we couldn't really think or imagine what the competitive advantage would be to having a little bit of phospholipid in a membrane. And so Itai just went and looked at it experimentally. And what he found, I think, was quite remarkable, which is that by making vesicles with mixed membranes, mostly fatty acids, but a little bit of phospholipid, they make perfectly normal vesicles like you see at this 0 time point. And they're stable and nothing happens as long as the whole population looks like this. But if they're in an environment where there are lots of vesicles that are made that have only fatty acids, you immediately start to see dynamic changes. And that occurs again because fatty acids can enter and leave membranes very quickly. And so they can leave the pure fatty acid vesicles and enter the vesicles that contain phospholipids, and these ones grow. And they grow over a period of minutes, again, into long filaments, as you can see in the time course at the bottom. So again, there's a way in which growth could occur by competition. And so we imagine that this could occur if there's some vesicle that evolves an RNA with the catalytic ability to make phospholipids, or perhaps other molecules with the same properties. And this changes the physical chemistry of the membrane such that that fatty acids prefer to be in this membrane than that membrane. And the physical chemistry behind that, I think we understand it very well, and I'm happy to talk about it later on, but I don't have time to go into those details right now. The important point is that here again, we have competition, right? So vesicles or protocells that evolve the ability to make phospholipids can grow at the expense of neighboring vesicles that don't have that ability. And we think that's very important because it would trigger an evolutionary arms race in the sense that to keep doing this, to keep growing by eating essentially neighboring vesicles that have less phospholipid, there'd be selective pressure to make more and more phospholipids. That would drive the population over time in the direction of what we see in modern cells, which is very stable phospholipid-dominated membranes. But on the way there, the properties of the membrane start to change. They become less permeable. And as a result, if you have other ribozymes that are carrying out metabolic reactions on the inside of the cell, that would start to have an evolutionary advantage, a selective advantage, because if you're making some valuable product on the inside, at the beginning, in a fatty acid vesicle, they just leak out and feed your neighbors. But as the membrane becomes less permeable, it would actually become advantageous to have an internal metabolism. And similarly, as the membrane becomes more and more dominated by phospholipids, it will become necessary to evolve transport machinery, probably peptide-based at the beginning. But again, you can see there might be selective pressures to evolve peptides and then more complicated proteins that can mediate and control transport of everything from ions to substrates and products across the membrane. And maybe the changing membrane composition is what actually drove the evolution of coded protein synthesis. Okay. So at this point, we can see at least a few ways in which primitive membranes can grow and divide and give us a primitive cell cycle. We're starting to see the chemistry of template copying, and we're working on getting a full cycle of RNA replication. And we try to put these things together and to make somewhat more complicated experimental systems that are closer to our picture of what a real protocell would look like. And I'm just diagramming here one experiment along those lines that was done by Kate Adamala when she was a student in the lab. And we've continued to do this kind of experiment to make sure that everything can work together. So what Kate showed is that we actually can add magnesium and nucleotides to the outside of these fatty acid vesicles. They can cross the membrane, get to the inside, and template copying on the inside can be observed. So it's just a little step in the direction of having RNA replication going on inside replicating vesicles. And we think if we could get real indefinite cycles of RNA replication inside vesicles that can themselves grow and divide and replicate, you would essentially have a system that where evolution can start to happen. Okay, so that's the goal of our work. Coming back to our picture of what a primitive cell or protocell looks like, we've started to realize that our initial ideas were maybe overly simplistic. We're thinking about the kinds of strands of materials, the mixtures that would be on the inside. We're thinking about how to make things compatible with the ionic environment, in particular, things like divalent cations, that magnesium that we need for RNA chemistry and which tend to disrupt membranes. We're trying to understand how to solve that issue of compatibility. We're thinking about ways in which simple peptides might have played roles. I can't say that we've made very much progress on that so far, but liquid-liquid phase separation of droplets might be a way to have local environments where things are more concentrated. We also know that peptides and even fatty acids can make filamentous structures that may play a role. So there's really a lot of interesting aspects to explore as we try to build these systems up experimentally. So let me just end with some of the big remaining questions in this field. One of the things I've really enjoyed a lot in recent years is the growing effort in the field to understand the geochemical setting that nurtured the first forms of life. In other words, what was the geological environment? Or should we be thinking about impact craters, volcanic environments, lakes, ponds on the surface, and so on, that nurtured life? I think one of the big and very interesting challenges for the future is understanding how the prebiotic chemistry that originally happened in the external environment somehow morphed into the biosynthetic pathways on the inside of cells that allow cells to become independent of the details of the particular environment that they're in. And of course, one of the long-standing and beautiful challenges is to understand how RNA catalyzed protein synthesis and the genetic code emerged. So see, these are some of the huge problems that I think people in the field are thinking about these days. And so let me just end by thanking all of the people that actually did all of this work. So I'm just showing you here some of the current and recent graduate students in the lab who've really made huge advances. So I think I mentioned Travis and Chris, but all of these people have made huge advances in our experimental and conceptual work. And over the decades, lots and lots and lots of people have contributed to different areas in terms of chemistry, reaction kinetics, RNA copying, membrane biophysics, crystallography, and theoretical approaches, and so on. And then I just have to mention that we couldn't have done all of this or any of this without support from all of these funding agencies. So thank them. And finally, just thank you all for listening to the story. And I'm very happy to take questions at this point. Okay, so thank you very much. And I think I'll stop sharing the screen so that we can see everybody here. So thank you very much.
Okay, thanks, Jack, for this wonderful talk. And we are now organizing a little bit the debate and the dialogue with you, with many questions from students and other people that could raise their hands and ask for a turn in the question part of this lecture. We have collected some questions by the students of the master. So Kepa and me will chair this session, and we will start with these questions by the students, and then other people attending can just raise their hand or write the question in the chat that we have. So the first question comes from Lydia, one of our students, and it's something related to one very nice thing that we were explaining about this unexpected way of dividing the vesicles. That it is very, very nice. So Lydia is interested in knowing which is the biggest surprise you came across when studying origin of life. In addition to that, you have any other moment of big surprise, of unexpected result?
You know, there's really been a lot of surprises along the way, but I think the biggest surprise was this growth of vesicles into filaments on the membrane side of things, and then figuring out the actual mechanism of the RNA copying chemistry and this totally unexpected covalent intermediate. That was also a huge surprise. But there have been a lot of other things along the way, and I think what that makes me what I want to say about that is that a lot of the advances in this field, and I'm talking not just about my work, but many people in this field, the advances are not usually limited by technology or sometimes even by experiments, but just by the way we think about things. And we get locked into preconceptions about how things must have worked, and then they turn out to be wrong, right? And we have to like rethink everything in light of new observations. So I think the field is very good for making us think in a very creative and flexible way, because our preconceptions are usually wrong.
I agree completely that preconceptions and prejudices are always influencing the work. Yeah. So Kepa, more questions.
One coming from Monica, another student, who asks about something that you also touched upon during your talk. This idea that there are in principle, a lot of molecular analogs or alternative mechanisms which are compatible with life, and this has been explored by many labs in the world. Mm-hmm. But she says that whether you think that this is the one possibility. So this is in the end we have just DNA, RNA, and proteins. This system that whether you think that this is just the one possibility, or is it the optimal one, or is it just a good enough one, or what this kind of question? Yeah,
that's a great question. It's one I think we probably all think about a lot, and I think there are huge debates in the field. So, I think there are some people who really think that from what we've learned about the chemistry, and including some of the things I talked about, that it looks like everything just points to RNA, and if RNA really is like the best, or maybe even the only way to get started, that I don't know. I don't really like that idea. I mean, intuitively, it just seems like it's more, it would be more likely that there should either be lots of ways or zero ways. And we know there's at least one way, right? So why shouldn't there just be lots of ways? It would just seem very weird if there was only one way to get to chemistry by which life could get started. And yet that is kind of what we're seeing so far. And of course, the obvious answer to that is, well, almost everything that's been done has been to try to understand how we got to RNA. So there's this whole space of other genetic polymers that has been, barely explored. And so there could be other ways out there. I think it's still possible that maybe in different environments where the chemical environment or geological environment was a little different, maybe there's a path to something else, or maybe not. I think this is a challenge to the field. It's just going to take a lot of time and effort to explore the space of genetic polymers and actually see if there are alternative pathways to life. It's a really central question.
It's a big question.
Yeah.
So one advice for the students: if you turn on your camera, we will see your faces, and it will be more living environment. Hector has proposed a question that is related to your work on self-replicating molecules and self-reproducing vesicles, but I guess you didn't pronounce the term synthetic biology in your talk. And how do you put your work or in the context of this synthetic biology movement or different approaches to artificial life?
So I can talk about that. So I think synthetic biology is it's almost come to mean two different things, right? So I do think that what we're doing is in a very real sense is synthetic biology, because we're trying to synthesize biology starting from chemistry. But more commonly these days, synthetic biology means taking existing aspects of biology and manipulating it, and either trying to build new kinds of structures, maybe taking different sort of subsets of what we see in existing cells and using that to make artificial systems that can do things that are useful for us in one way or another, or just as a tool for understanding how cells and tissues work and form. So that's also, it's just a different conception of synthetic biology. But I think the common thread is that we're all trying to build things that are biological in different ways.
Okay, thank you very much.
Okay, a question coming from Alvaro, another student, who says, at the beginning of a project like this, I suppose that when you were starting to face the origins of life problem, how do you tackle the design of essays and experiments to answer the questions that are so difficult or so big?
Right.
How do you go, step by step, or how do you do that?
I think that's also a great question, which really applies to any big question in science. If you just say origin of life, it's kind of like, oh my God, where do we start, right?
Sure.
So any big problem, you have to think about it, try to break it down into smaller chunks. Those chunks themselves might be very big problems that you, again, can break down into smaller questions until you get to a point where you can actually do a realistic experiment in the lab. And so, so we have our simple conception of a protocell. We start off by, in a sense, separately looking at membrane growth and division, and separately looking at the formation and copying and replication of RNA. And in that copying chemistry, we can break it down into all of the chemical steps. And you know, similarly, you just have to break these big questions down into smaller bits and pieces that are something that can actually be tackled both theoretically and experimentally. And of course, the problem with that is that you end up with lots and lots and lots of little pieces, right? So it ends up being like, to get back to solving the big question, it's like putting a jigsaw puzzle together. You have all these pieces and you have to ask, well, you know, what fits with what? You know, is this piece over here actually, you know, doesn't fit anywhere, and we have to go back and look for alternatives. And so a big problem like Origin of Life, you know, you can't solve all of the pieces together at one time. And so we, in a sense, we have to be satisfied when we solve little individual bits of the puzzle. And of course, it's always nice and beautiful when we see, oh, now we can have 3 or 4 or 5 pieces that actually fit together in a nice way. And that's what makes us think that we're getting closer to a realistic understanding.
Okay, there are also several questions proposed by Lydia, Carla, Laura, Karen, that are more related with the change in your career from biomedical research related to telomeres and senescence, cell senescence, to the field of origins of life. But one question were related to what were the motivations of changing to that field, and also the difficulties. I remember now a conversation with you some years ago with Marie-Christine Morel, having lunch in Paris.
Mm-hmm.
We're explaining us, that was before the Nobel Prize, okay? Mm-hmm. The difficulties to explain to some funding agencies about the usefulness of your research. So could you explain a little bit to the students about this change because it seems it's a very radical change.
It is, in a sense, a radical change, but I guess I'd point out that it wasn't a sudden change. We actually made the switch over a period of many years. And it's actually not even that radical a change, because a lot of what we were doing, especially in the earlier days, was more or less molecular biology or simple biophysics. Over the last 20 or 30 years, those small changes have added up to a pretty radical change. So in a sense, of gone from being a biologist to a chemist, and I'm actually joining the chemistry department at UChicago. But yeah, it's really driven, I think, by just interest and curiosity about how different things work. So even back when I was more of a biologist, I've studied recombination for many years and double-strand break repair and things like that. And the getting into the telomere field was really triggered by a conversation with Liz Blackburn, and just the idea that, wow, there's like something really interesting here, and there are simple experiments that we could do to contribute to the field. And so in the early and mid 80s, when ribozymes were first discovered, I kind of had the same feeling that, there's really a lot of interesting questions here, and maybe not that many people looking at them. So I thought there would be some way that we could contribute to the field. And it's been so interesting and so full of questions that it's just driven me more and deeper into this set of puzzles. In terms of funding agencies, that is important. And one of my frustrations with the way that most funding agencies work is that they make it very difficult for people to change fields, right? If you've worked in one area for many years and then you make a proposal to do something in a different area and that gets reviewed by committees, and then the typical reaction is, well, what is that? What does he or she know about this new field? They've never done anything in that area, and so then it's not funded. But we all know it didn't take us very long to become experts in whatever our particular subfield is. It's not that hard to learn a new field. It's certainly not that hard to start doing something interesting if there's an approachable question in a different area. So I think the funding structures, really a lot of attention needs to be paid to finding a different model that encourages people to explore new, interesting questions and encourages people to be creative. And I've been very fortunate to be a Howard Hughes investigator for many years now. And I like their model of just, they try to find people who've been productive in one area and then just support them to do whatever they want, right? So it's basically the people, not projects model. And I'm not saying that all science should be funded that way, but I definitely think that science would progress more rapidly if we use that kind of model more widely, so that people have the freedom to explore new questions.
That would be very nice.
Okay, a couple of. I'm gonna, if you don't mind, Julie, I'm gonna merge two questions, 7 and 9 here. In the same vein, related more to a career, your career pathway. And so one says, from Karen, says, what would be your advice to have a successful career like you have had? Kind of the main advice that you would give to a young researcher. And the other one is from Sarah, saying, how do you deal with failure when there is failure in the some unexpected results that really don't look at all promising, or things like that? It's something that you cannot integrate with anything else. And
right.
So the two things together. They were two separate questions, but I think that they make sense together as well.
Yeah, they're very related. And I have to say that science is hard and failure is normal, right? So if something doesn't work, it's not the end of the world. It just means maybe you need to think about it in a different way, or just switch and do something else. So and in terms of getting started, I think maybe the most helpful thing might be to just tell you that, as a graduate student, I had a really terrible time. It was really a very, very hard time in my life. And part of that was just because of in retrospect, some mistakes that I made. So I got very attached to trying to solve a scientific question, which, was a very interesting scientific question, but I ended up in a lab. So I wanted to take a genetic approach to simple developmental biology questions. And I ended up in a lab where there was no expertise in genetics. So I didn't really have anyone to talk to. So I think it's just super important to be in the right kind of environment and to be in an area where you can ask people for help. You can ask questions, you can get advice, you can find expertise to solve a problem. And so even when I finally gave up on that project and I actually moved to a different lab, almost the same thing happened again. I had come up with, I think, what was a very cool project for the time. I mean, it sounds almost silly now, but the idea was to make a short DNA oligonucleotide and use it as a probe to detect a particular gene or message, which at the time took years. And the lab that I was in did not really have the expertise to do that. And my advisor, Ray Wu, eventually recognized that, and he rescued me by saying, look, Jack, you got to learn how to do this properly. I'm going to arrange for you to go and visit another lab, learn how to do this chemistry, come back, And so I did, and it just really brings home the point that, that there's, if you don't have the right expertise in-house, or you don't have the right people to talk to, you gotta reach out and get help. And we solve these problems not usually in isolation, but by talking to each other.
That's really nice. Good advice. Yeah, indeed.
Okay, Kepa, we don't have more.
No, there's one that was in between the two that I merged. It was what application from your discovery are you most proud of? This was in principle the one that you had to do, Julie, but I do it myself.
Okay.
So what are you most proud of after your,
Well, let's see. So I'm not really expecting applications to come out of what we're doing now in Origin of Life, but earlier when we were developing in vitro selection technologies and trying to understand how to get functional molecules out of random sequences, we spent quite a bit of time working on a kind of complicated and difficult technology called mRNA display. It was basically a way of displaying newly synthesized protein chains and having them attached chemically to their messenger RNA. And that allows you to go through a cycle of selection for the protein to do something, and yet being able to amplify and enrich for those sequences. And so we thought that would have some practical applications. So started a company, we did all kinds of great science, and then ran out of money and went bankrupt, and then thought about it for a few more years, and then got together with Doug Trico, who was a postdoc in the lab 20 years earlier, who'd since become a biotech entrepreneur. And we tried again and started another company, and that company actually learned how to, well, sort of built on work from my lab and other labs, learned how to make highly modified cyclic peptides and evolve them. And that has actually led to a drug that's in clinical trials. So to see something go all the way from really basic science and a technology that lets you develop a new drug that's actually being used to treat people, that's been very satisfying. The other thing that's been super interesting to me is that just because I've worked on nucleic acids my whole career, I ended up getting involved in a biotech company you've probably heard of called Moderna, which is one of the ones making these mRNA vaccines. And so, I don't claim to have contributed anything significant to the vaccine story, but just being able to watch the progress from, again, very basic science all the way to vaccines that have probably saved millions of lives has really been amazing. So you never know when, something useful will come out of either your work, or maybe you have the opportunity to watch and help something else practical come along. It's very nice. So, even though the work that I'm doing right now and the work in my lab is very basic and we don't see applications, there are still opportunities to work in areas that are more applied. And I love being involved in biotech companies and watching and helping new ideas come to fruition. It's a different way of doing science, but it's also very satisfying.
Thank you very much. I think that they should be satisfied. The students should be satisfied.
Okay, before giving the floor to the questions that are coming from the YouTube channel, anybody in the room, Carlos, Kepa, the students, maybe they can propose a new question. Other people, Carlos, you.
Yeah, I have a question first, thanks for your very, very nice talk, Jack.
Thank you, Carlos.
Lots of space about the RNA world. That is one of our favorite issues in science, as you know. And I have a question regarding this amazing transition between spherical shaped protocells into these linear shapes you can see using the microscope. Have you been able to measure any trend towards more, let's say, catalytic activity of these structures because of the higher, I mean, the increase in the ratio between surface and the volume, and also the possibility of stacking those structures and promoting, for instance, polymerization of monomers both inside and outside the protocell structures?
Yeah, we've tried to look at questions like that. We have a few papers and ways in which you can, we've tried to localize RNA molecules to the membrane surface with exactly that in mind, that, what's a three-dimensional search problem in solution is now a two-dimensional search problem on a surface. So it might be a way to speed up reactions. And I can't say that we've found anything very effective. Maybe we're just not doing it in the right way or looking at it in the right way. It's a very attractive idea, but we haven't gotten very far.
That's why I'm asking it, because I've never read something from your lab telling that some increase in this catalytic activity has been followed during this amazing process. That perhaps one should expect that those lamellar structures could contribute to at least polymerization of monomers.
No, I think there's definitely something interesting there, and there's been a few ideas like you can assemble ribozymes from the non-covalent assembly of fragments, right? And that might be something that if they're all localized to the surface, could be much more effective. But having that localization chemistry in the first place, it's not really clear how to do that. One thing we're thinking about is maybe there's a connection to amino isolation chemistry.
Okay, this gives me gives me a good good entry into my question that I wanted to ask about both amino acids, peptides, and metabolism. Okay, you give us a general view in which the driver initially is the environment, and the end of the whole process is our genetically instructed metabolisms. Okay, and we in the RNA hypothesis, you ascribe a lot of let's say causal power to a molecule that doesn't have that much causal power, as you were also saying during your talk. There are, I mean, RNA is everywhere, as you were saying, but it's proteins, as you said, that do most of the jobs. And this search for the replicator, when nature doesn't have a self-replicator in the cells, could be kind of a human obsession or I mean, you know what I mean? And the place of metabolism and origins of metabolism in your picture, because that's the part that somehow comes out of natural selection acting on these replicating molecules, but it's not so clear how it gets embedded, and all the problems of synthesis and stuff like that.
Let me say that my way of looking at this has changed a fair bit over the years. So yeah, we used to be obsessed, I think, with maybe the first ribozyme being a replicase, right? And so it's all RNA centric, and we're all talking about. But I think, kind of as you're suggesting, what's the point of having a self-replicating RNA if it's not connected to anything else, right? And now, sort of going back to Itai's experiments on the membranes with phospholipids, I think it may be more likely that the first ribozyme that evolved is actually doing some metabolic job, like, for example, making 2-chain phospholipids. And only once you have a useful ribosome is there a selective advantage for replicating it more efficiently. And so they become very coupled processes at that point. So that's one aspect. The other thing that we've been thinking about lately is a little bit on the origin of protein synthesis, and the substrates for the ribosome are amino-acylated RNAs, right? They're RNAs with an amino acid at the end. But how could you evolve coded protein synthesis if you didn't already have those substrates, right? Sure. We don't have terrific chemistry for making them. So what we've been trying to think of in the lab are, are there potential functions for amino-acylated RNAs that came before the evolution of the ribosome? And if so, that might drive the evolution of ribozymes that are amino-acyl RNA synthesis. But what would that primordial function be for an RNA that has an amino acid at its end? So that's an area that we're actively looking at. We have one paper out on that and another just recently submitted. So, we're working on it. I think there are a lot of possibilities, but we need to think about it a lot.
Yeah, I would mention Robert Pascal's work on the amino acid chemistry and nucleotide chemistry, how they could, from the bottom up, how they could come together. And from a top-down approach, the Loren Williams approach to the structural, the evolutionary development of the structure of the ribosome also points to the let's say, the coevolution of amino acid peptides and oligonucleotides rather than maybe just amino acids and a big RNA molecule. So there are lots of things to explore there, I think.
It's clearly a long process of coevolution. And I think this also feeds into the Sutherland chemistry where we see both amino acids and nucleotides and lipids coming out of the same underlying chemistry. And the question is, how did they get together? And what was the pathway?
Sure.
And also in this field, this current claim that even a single nucleotide bound to an amino acid can promote, let's say, peptidyl transferase or amino acid transferase has been published this summer.
Yes, right. I mean, there's no coding in there, and I know it's an interesting step, but I think there has to be something that is more directly connected to the way peptide synthesis works nowadays.
The genetic code in that system is only 4 amino acids long. I mean, you can code for and they use only 4 amino acids.
But it's done artificially, right? You can put a different amino acid on different nucleotides, but how would that actually happen in a primitive cell? Again, maybe by ribosome or something.
Okay, Carlos,
we should refrain ourselves.
This is for to go on all day.
Otherwise, we will tire out.
Questions coming from the YouTube channel, and Germán will present these questions in order to not to be the session too much. Germán.
Yes, I'm here.
Okay.
Well, it's time to ask Professor Szostak a few questions coming from the audience following the talk through the CSIC YouTube channel. And I'd like to thank all of the people attending this event through this channel. Because of time constraints, I just selected a couple of them, and the rest, as we agreed yesterday, we will make a list of homework for Professor Szostak, including some from my own that probably were a little technical for today. And we will, I'm pretty sure that the you will be happy to at least get the questions, and then if you get the time, you can answer them. I'm going to, the first one of the two that I selected is why simple fatty acids are transferred preferentially to phospholipid-enriched vesicles. Is the charge of the phosphogroups, or is something more physical or mechanical?
Yeah, we think there may be at least two reasons why membranes that contain some phospholipid tend to grow by integrating, by attracting more fatty acids. So the underlying phenomenon, well, one thing that's very clear is that it's a difference in the rate at which fatty acids in the membrane come out of the membrane, the dissociation rate or the off rate. And that seems to trace back to an increase in order in the membrane that is imposed by the intrinsically higher order of the two-chain phospholipids. And so we can measure on and off rates. The on rates are the same. The off rates are slower if you have phospholipids, and so they tend to grow. There's also a simple entropic mechanism. So as the membrane grows, by absorbing more fatty acids, the phospholipids are diluted, and that's entropically favored. So there's a combination of a biophysical property of order in the membrane with this entropic effect, which we think together explains the growth phenomenon.
Okay, thank you very much. And the last question is from Felix Campello in Barcelona. And the question is, how do you think those protocells divide? Would the addition of trace amounts of complex phospholipids help divide the cell, perhaps by increasing area to volume ratio upon growth?
So if you go through this pathway making long filamentous vesicles, they're very easy to divide, but it requires environmental fluctuation, some kind of turbulence, which you can imagine. I like to think of, some wind blowing over the surface of a pond, you get waves, and that's enough, generates enough shear forces to cause these filaments to fragment. But we have actually much more recent work done by Anna Huang when she was in my lab, and more recently in her independent position in Australia. Anna's worked out how to make large vesicles with a single membrane. And when those grow, so the surface area increases, then they spontaneously start to have very large scale fluctuations, which lead to budding and division. So there are different pathways that lead to growth. And in those experiments, I don't think we've yet looked at the influence of phospholipids. So it is a very interesting point, would there be differences in membrane composition, or perhaps the addition of molecules that might affect the energy of curvature of the membrane that might either trigger or inhibit division. So there's a lot to explore there.
Okay, thank you very much. We will send you the rest of the questions. And just to finish, let me say a few words to conclude this event. On behalf of the entire team of the Master in Integrative Synthetic Biology, that, as Julie said, is co-organized by the Spanish Research Council and the Menendez Pelayo International University. With great enthusiasm and effort, all of us are making possible this advanced school, which is a collective higher education action of leading CSIC centers working on complementary aspects of synthetic biology and related disciplines. Thank you, Professor Szostak, for accepting our invitation to deliver the opening lecture of our school, that is the first one of this kind in Spain. The mission of this school is to prepare the next generation of synthetic biologists represented in the Zoom room by the MISB students to explore with novel ways molecular and cellular systems. In other words, engineering to understand and master biological complexity. Your talk on the origin of cellular life fits very nicely with the scope of the school, as mastering the intrinsic capabilities of molecular and cellular systems will contribute to our better understanding of the basic principles of life and its emergency from lifeless components, which, as you said, is a grand scientific challenge of this century. Moreover, all this knowledge and efforts will provide powerful tools for novel solutions to outstanding environmental and health-related problems. I'm confident that your master lesson will impact the future in science and perhaps in life of part of the young audience who has just been listening to you. In last year's opening lecture of the previous master school that delivered by Professor Bruce Alberts, I concluded with the following words that I consider pertinent for today's closing. Dear students, as scientists and teachers, we will try to do our best to help you keep the momentum. And if you finally decided to continue exploring the unknown, becoming a scientist, please don't forget that science is more than just a profession. It is a way to look at yourself and the world around you and solve problems that matter the answers to science generally or humankind. Thank you all for your attention and for participating in this event. With our gratitude, Professor Sostak. From now on, please consider the Spanish Research Council your home, or at least one of your homes in Spain.
Thank you very much, Herman. Thank you, everybody, for inviting me to talk here. And I think your program is going to be very successful. Really appreciate the great questions from all the students. So I think you have great students. They're all going to do great things. So yeah, it's been a privilege to talk to you all today.
Thank you very much.
Okay, so it's bye-bye. Julie, you should say bye-bye, no? Something like that.
Well, no, we are going to close now the YouTube channel, and the idea is that we can say goodbye in a more private way.
In a more private way. Okay. I stay here then, just for a few minutes.
You can make him, you can